Hey, so in today's lesson, we're going to look at dihybrid crosses, 9331 and the test cross. So this is part of the higher level IB curriculum. We will focus just on dihybrid crosses today, and then I'll make a follow-up video looking at linked genes and how that works with chi-squared. So this is an interactive lesson that you can pull from Biology by Bradford and have a look back through. First of all, we've already covered and hopefully watched the video about monohybrid crosses, and that's where we're looking at the inheritance of one characteristic, where it's either dominant or recessive. So we have our classic two heterozygous across together, and we get a 3-to-1 ratio. What you'll see here is that we're looking at two traits. They're inherited separate from each other. So this is representing different chromosomes, and we've got our capital letter R is our dominant gene, and our lowercase letter R is our recessive gene. And the idea is that they are following the law of independent assortment from Mendel. That means that they are going to find themselves in either one of these combinations, or also there's obviously a third one as well, where if these ones were the ones switching around, and whether or not we get this chromosome or that chromosome from the pair is completely independent of what happens with these guys here. So that means that they are inherited separately, and that the inheritance and the assortment of one does not affect the other. So independent assortment. When we say assort, we mean like the way that they find themselves in this arrangement here. So the other thing to think about is that we're looking at a dominant and a recessive trait of two different characteristics. And there's the typical classic two heterozygous individuals across together, and we end up with a 9:3:1 ratio. So the other ratio I said to remember for monohybrid crosses is a 3-to-1. Here we want to remember 9:3:1. And if we take our gametes, the possible gametes from a heterozygous individual, we get big R, big T, big R, little r, big T, and little r, little t. And that will be the same for both. And what you can see is that we're going to have 9 individuals that have a dominant trait for both. So they're round and tall. If we're looking at that as our dominant trait, round being dominant to wrinkled, and tall being dominant to being a dwarf, like small. So we're looking at a plant here. It's the easiest to do with plants and to figure out these ratios, because we're not only going to look at 16 individuals in order to get this ratio, we're going to have to look at thousands, because it's just an average. And, like if you're trying to do statistics on just 16, then by chance you'll have a different ratio to this 9:3:1. And then when I say 16, because 9 plus 3 plus 3 plus 1 is obviously 16. And then 3 of them are going to be round and a dwarf. So that means they have a dominant trait. As you can see here, they've got the dominant R coded for trait, which is round. And then they are a dwarf. They have the homozygous recessive for height. And then the inverse of that would be homozygous recessive for size, which means they're for shape, which means they're wrinkled. And then having the dominant trait of being tall. And then one individual is homozygous recessive for both traits. So we want to remember 9:3:1. So if we just skip down to like an example problem. So maybe you get asked in tomato variety, tall stems are dominant to dwarf and hairy stems are dominant to smooth. The two genes are unlinked. So that means that they are inherited independent of each other. When we come on to talk about linked genes, it is what it says on the tin. It means the two genes will be inherited together. They're on the same chromosome. So when they're unlinked and they're on separate chromosomes, that's when they're going to follow this inheritance pattern. And two plants that are heterozygous with both across determine the expected phenotype ratio. If you see the word determined, it means to show it as a Punnett square. And we've got step 1, figure out the alleles. Step 2, what are the parents? Step 3, we want to know what the gametes are. And we can do that by foiling. And then step 4, we use our grid and then we'll count them up. And what I mean by foiling is if we look at this individual here. Let's go up to the heterozygous one here. If we take the first, so from the big R, big T, the outside, the big R, little t, the inside, little r, big T, and the last, which is the last of this pair, the little r and the little t. That's foiling. So first, outside, inside, last of our like parental phenotype, genotype. Now, the other thing to think about would be, what is a test cross? So, like, what is the outcome of test cross? And these are the other kind of problems that you might have. And then when we come on to linked genes, that's important to determine whether or not any difference we see is due to chance or if because genes are linked. So when they're not linked, what we're expecting is that if we have, so here is our tester, so it's a homozygous recessive, it's our tester because we can see the phenotype, of course, that it's recessive for both traits. So we know what the genotype must be. There isn't a possibility of it being heterozygous for one trait or homozygous dominant. It's not possible. They can only be homozygous recessive for each trait. So that's why we use a homozygous recessive individual as our tester, because we know what the genotype is based on its phenotype. Whereas if you are like an individual like this heterozygous, like this one here, or homozygous dominant, we don't know, like looking at you, what is your genotype, because you have the same phenotype. And then, so if I do a test cross with a homozygous recessive, and I take someone that is homozygous dominant, they will all be heterozygous. And if I take someone that is heterozygous, and cross them with someone that's homozygous recessive, I'm going to get a 1-1-1-1 ratio. You'll probably remember when we do a test cross, when it's a monohybrid cross, we get a 1-to-1 ratio. And now this time, just like with the heterozygous individuals being crossed together, we've got 4 possible outcomes, and that's a 1-1-1-1 ratio. So memorize those 2 ratios, 9-3-3-1, so 9-3-3-1, and memorize this 1-1-1-1 ratio. There are some other ones like this where you might have a 1-to-1 ratio, but I've never really seen it tested. It's worth being aware of it. If you've got 2 traits, then the outcome of that would be that one of them is heterozygous for one of the traits and homozygous dominant for the other trait. So at the end of this interactive lesson, we've got right down the bottom, this is what we're going to be doing next lesson. What are linked genes? And then how can we use a test cross to figure out whether or not genes are linked, and how does chi-squared help us determine this? I will do a separate video for that because it's something that stumps a few people. And then if you go down to the bottom, we've got some multiple choice questions. So let's do a couple together first. How many different gamete types can a plant of a genotype that's heterozygous produce, assuming the genes are unlinked? So you probably remember that would be 4 outcomes. And this is foiling it. First, big R, big T. O, so outside, would be this one. Little r, big R, little t. So that one. Inside is our little r, big T. And last, little r, little t. So foiled it. What cross is expected from a 9-3-3-1 ratio? 2 heterozygous individuals. Now, unfortunately, the IB questions will not be as simple as this, but it kind of gets you in that frame of mind and helps you memorize those facts. So in F2, that means the second generation of this. What proportion of offspring are expected to be both wrinkled and dwarfed? So wrinkled and dwarfed is homozygous recessive for both. We just have to think back to that previous information. There wasn't much context for that question. And which genotype appears most often among the 16 boxes so out of 16 individuals, Two heterozygous individuals crossed together. What one are we going to find the most? A heterozygous individual. We could go up and quickly count that, but it's always fun to just have a random guess. So what I'd like you to take away from today is that 9:3:3:1 ratio. You can also see it here, 9:3:3:1. And also when you do a test cross, you're going to get an individual. If it is homozygous recessive is our tester, and that's going with a heterozygous, we get 1:1:1:1 ratio. And if it's homozygous dominant, then they're all going to be the same. So remember to check out, if you haven't done so yet, my free study page, which is cramley.study. And there's a whole bunch of little interactive games that are available there as well. And you can just log in, make a free account. And this is spaced repetition to help you memorize all the jargon of IB biology, and also some application style questions to help you prepare for any test. Okay, I hope you found it useful today. Remember to like, subscribe, and share. Thank you.